It is my great pleasure to welcome you to this symposium. I am very happy to organize this symposium, which will focus on multimodal analysis of neurodevelopment and function. Let me give a brief introduction of this forum. The complexity and function of the nervous system relies on the generation of unparalleled neuronal diversity across molecular, morphological, functional, and connectional features. Diverse neuronal complexes coordinate with glia cells to form a complicated neural network to exert physiological functions. Thus, multimodal analysis of the molecular profile, fate decision, morphogenesis, neuronal activity, and circuit assembly of neural cells is fundamentally important for us to understand the brain development, structure, and function. This forum will introduce how to employ single-cell multi-omics brain-wide connection of neuronal morphology, single-cell resolution calcium imaging, and high-resolution connectome mapping to investigate the fates of neurons and glia, as well as neuroregulation of survival behaviors, puberty, and social behaviors. So let's kick off the symposium. Today, we will have quite a few talented researchers with us to give talks in this specific symposium. The first speaker is Gao Zhihua from Zhejiang University. She will talk about oxytocin neurons, emotional control, and social behaviors. So let's welcome Zhihua Gao, professor.
Can you see me and hear me?
Yes.
Okay, yeah. Thank you very much, Qing Feng, for organizing this meeting, and thank you very much for the invitation. Today, I'm going to share with you some of our findings regarding the oxytocin neurons, and particularly the neuroendocrine oxytocin cells that are involved in emotional control and social behaviors. So please allow me to, minimize. So oxytocin as a hormone, it has a long history, more than 120 years. So it was originally found as an extract from the posterior pituitary in 100 years ago. And at that time, most of the studies have focused on their roles as to promote uterus contraction and facilitate milk ejection. And then during the last 40 years, most studies actually focused on its role as a neuropeptide in the brain, because oxytocin has been shown involved in maternal behavior, social behavior, pair bonding, and other emotional controls. And we know that oxytocin is primarily produced by neurons located in the hypothalamus, particularly in two main nuclei, paraventricular nucleus and supraoptic nuclei. And these neurons directly project to the posterior pituitary over there. They secrete oxytocin into the peripheral bloodstream, and then they get transported to distal organs to regulate parturition and lactation. As I just mentioned, recent focus have been working on its role in emotional and social behaviors. And this is related to the released oxytocin in the central brain as a neuropeptide. So I'd like to draw your attention is that oxytocin neurons, while they're primarily located in the paraventricular nucleus and the supraoptic nuclei, there are actually two types of oxytocin neurons in the paraventricular nucleus. For example, the primary type is the magnal oxytocin neuron that I've just introduced to you. They project to the posterior pituitary, and they are bigger in about 25 to 40 micron in diameter. And these are the neuroendocrine cells, and they release their hormone directly into the blood vascular secretion. While as in the paraventricular nucleus, there is another type of oxytocin neurons that have been identified a number of years ago by Swanson's lab. Those neurons, they primarily project to the brainstem spinal cord, the hindbrain region, and they are smaller with like 15 to 20 micron in diameter. They are not the classical neuroendocrine cells. They release oxytocin in the brain. So the question is, does the central action of oxytocin neuron, which type of neurons actually mediate its central actions? There were quite a few studies focusing on this, but today I'd like to show you that micronal oxytocin neuroendocrine cells, they do also involve in the central actions of the neuropeptides by their collateral projection. So let me first introduce the. I'll have like my talk will be divided into two main parts, and the first part I'd like to show you that those micronal oxytocin neurons have collateral projections to different brain regions, and by doing this, they facilitate social behaviors. So as I just mentioned, microneural endocrine cells, they project to the posterior pituitary. However, in order to selectively manipulate this population of cells has been a big challenge. So over 40 years ago, scientists have been trying to label the cells using classic HRP-based retrograde tracing from the posterior pituitary, and they were successful in labeling the cells. However, reconstructing the system and well-define their projection has been difficult. So in our hands, we used this high-efficient retrograde viral tracer by injecting this retrograde tracer into the posterior pituitary. We were able to trace the cells from the posterior along the neural stem back to the hypothalamus in different brain regions, such as the supraoptic nuclei and the paraventricular nuclei, as well as other brain nuclei in the hypothalamus. And these traced cells actually span over from the preoptic area to the mammalian area of the hypothalamus, so spanning quite a few layers. And as expected, they contain the majority of neuroendocrine cell peptides, vasopressin, and oxytocin. And this method actually does not label the parvocellular cells that project to the medial eminence, suggesting our strategy is selectively labeling only those neuroendocrine cell projecting to the posterior pituitary. And we then collaborated with Dr. Hui Gong's group using their fascinating FMOS strategy to combine, to fully reconstruct the whole brain projections of those cells. So by doing this, so we cut the brain into thousands of slices and use the fluorescence imaging to reconstruct all the cells that project to the posterior pituitary, and these cells resemble the ensemble of neuroendocrine cells located in paraventricular nucleus, supraoptic nucleus, as well as other areas. And this, so by maximizing their projection, we obtained their three-dimensional architecture of the system as shown here. So this really represents the beauty of our neuroendocrine system. Like there are hearts embedded in the projection and the butterfly structures that are located in the paraventricular nucleus and anterior commissural nucleus. And those are mainly, and the cells contain mainly oxytocin cells. I'd like to also draw your attention is that other than the cell somas, we also observed this long, thin processes And this process, because we traced the cells using those retrograde tracing strategy, this actually suggesting that all the fibers that we see in the brain region may well represent the collateral projections of those classical neuroendocrine cells. So as we've shown here, that the cells actually have fiber distributions in quite a few brain areas, such as amygdala, cortex putamen, piriform cortex, and these areas have been shown to be involved in oxytocin-mediated central actions, such as social reward, fear attenuation, and other like social recognitions. So do they really represent the collateral projections? We used the single cell fiber tracing, taking advantage of this, the high resolution of the fMOST imaging. So we did the single cell tracing and showing that here, that's one of the paraventricular nucleus cells project to the posterior pituitary as well as the nucleus accumbens. And by tracing more than 25 cells, we found that about 5% of cells do project collaterally project to different areas, such as the lateral LGP, CPU areas. And we then also reconstructed this video that shows the trajectory of those microneuroendocrine cells, that they do have these collateral projections in different areas, which and we also used the dual viral retrograde tracing by injecting retrograde traces either into the posterior pituitary or different brain regions, such as amygdala, cortex putamen, and nucleus accumbens. And we traced those cells that located in paraventricular nucleus, and most of them are indeed oxytocin cells. We then made a oxytocin-specific query line, used similar strategy to selectively label oxytocin magnocellular cells. As shown here, this method selectively labels those magno-oxytocin cells, but not the parvo-oxytocin cells, suggesting the specificity. And we also observed their fibers in different brain regions. So, and by doing this, we have a way to selectively label and potentially manipulate them. So to manipulate the cells, we injected those dread virus like M3DQ or M4DI virus into the posterior pituitary and selectively labeled the cells. And by CNO injection, we successfully activate this subpopulation of cells in SON and paraventricular nucleus in PVN. As shown here, in M3DQ infected cells, false CNO injection induces false expression as indicated by their activation in this particular type of cells. Whereas conversely, in M4DI expressing cells, CNO administration reduces the firing of the cells. Along with this, we were able to detect the change of plasma oxytocin levels in this mice. For example, CNO administration into the M3D infected mice increases the oxytocin level, whereas the other, conversely, M4DI infected animals shows reduced oxytocin levels. So how does it affect the social behaviors? We use the 3-chamber assays, and that, as we know, it usually the rat always prefer to interact with their conspecifics. So when we activate the magno-oxytocin neurons. This preference is significantly enhanced as shown with this increased chamber duration and sniffing. Conversely, if we suppress the magnocellular oxytocin neurons, this preference, the original social preference, is attenuated with the reduced duration and sniffing with the conspecifics, suggesting that this magno-oxytocin neurons not manipulating magno-oxytocin neurons not only change the peripheral plasma hormone level, but also affect their central social behavior. Since we observed the fibers in the caudate putamen, we also use this local infusion of the oxytocin antagonist into the caudate putamen, as shown here, that we notice that when we activate oxytocin neurons, rats shows increased locomotion. However, if we locally infuse antagonists, and those animals, the increased locomotion is inhibited. And we propose that this increased locomotion in the caudate putamen area may help to facilitate social approaching during social behaviors, as they have to approach their conspecifics to have their social interactions. So to summarize this, we reconstructed this three-dimensional architecture of this microneural endocrine cell system, and we found that oxytocin neurons have collateral projections to both the posterior pituitary and the central brain regions, and this actually promotes locomotion, may facilitate the social behaviors. So now I'm going to quickly go through this. The neuroendocrine oxytocin cells also regulate our emotion. As we know that stress always affect our endocrine system, and one of the most striking feature is that the activation of hypothalamus pituitary adrenal cortex axis. And when we are stressed out, the cortisol levels always increased, as also seen in depressed or anxiety patients. However, studies also notice that patients with depression or anxiety, they do quite often have reduced plasma oxytocin levels. And we found it in the chronic restraint-induced anxiety models, this cell, the animals also showed reduced oxytocin level. So we wonder whether directly manipulating oxytocin endocrine cells actually affect emotion. Although previous studies have shown oxytocin do inhibit negative emotion, it's not clear whether it's mediated by neuroendocrine cells. So we, to address this, we use this chronic restrained stress model as shown here. So 21 days of restrained stress were caused dramatic or apparent anxiety-like behaviors in rats. However, if we selectively activate oxytocin endocrine or magno-oxytocin neurons, we will be able to see that the reduced central entries or reduced locomotion, as well as the reduced open arm entry, has been very well rescued. And as well, this also is accompanied by elevated oxytocin levels, suggesting that this neuroendocrine system also directly participate in the emotional control. To our surprise is that because this anxiety model was made by 21 days of restrained stress. So we then wonder, like, if we give the animals a sub-threshold stress, do they still have an anxiety-like behaviors? So with the 7 days of restraint, we notice that the animals do not show an apparent anxiety phenotype. However, with the same period of oxytocin neuron inhibition, we do observe very dramatic anxiety-like behaviors. And how does this contribute to the anxiety? And that is still under investigation in our group right now. So to fully summarize this, that I've briefly introduced you about the oxytocin neuron system, and we focused on the magno-oxytocin endocrine neurons. And these endocrine neurons, their classical role is to project to the posterior pituitary to regulate lactation and parturition. However, we also show that these cells, they collaterally project the axons to different brain regions to regulate social behaviors and emotions. By doing this, they orchestrate their peripheral and central actions. And that's all. I'd like to thank you all for your attention, and I thank our funding agency, our collaborators, as well as our group member Bin Zhang, Li Yao, Wei Xiao. They've contributed a lot in our project. Our colleagues at University of Edinburgh and Amsterdam has helped us and support us in our journey to explore this neuroendocrine system. And thank you very much for your attention.
Any questions from the audience? Okay, here I have a very short questions. I am wondering whether there are both like magnocellular neurons and parvocellular neurons, oxytocin neurons in the SON. Because in the recent published paper from Xuetian's lab, they think that the oxytocin neurons in SON can project to the paraventricular nucleus, right? And there could be like reciprocal connection between the SON and PVN nucleus.
Yeah, this is a very good question. So I think we think most of cells in the SON are magnocellular neurons that directly project to the posterior pituitary. However, these cells probably also do have collateral projections to the paraventricular nuclei. And on the other hand, parvocellular cells in the paraventricular nuclei also project to the SON's magnocellular neurons. So they probably are like, we've just showed that magno-oxytocin neurons orchestrate peripheral and central action. There is another option that both parvo and magno-oxytocin neurons, they orchestrate their actions in the brain and the peripheral system.
Okay.
Zhihua, maybe I can ask a question directly. So this oxytocin neuron, do they also do this? I mean, does this, this neuron also can release this oxytocin to the blood?
Yes.
Yes. Go ahead.
They release their hormone, oxytocin, into the posterior pituitary over there. Those are the fenestrated capillary. So they get into the bloodstream.
So I'm just curious, because this neuron this peptide not only released to the blood, also they can, of course, they can accept by some target neuron. When you activate this neuron, so so in the central nervous system, of course, some neuron might be activated, or in the peripheral organ, they also have their target organ. So do you know which one is the major player for this behavior difference when you activate or inhibit this neuron?
So we think for the emotional behavior part, we think it's mainly mediated by the central actions of oxytocin, because once oxytocin is released into the bloodstream, it barely passes over the blood-brain barrier and re-enter the brain. So that's why. And in the peripheral organs, it is the uterus and mammary gland that express high levels of oxytocin receptors, and that's why they have this prominent effects in response to oxytocin stimulus.
Okay, great. Also, I saw the second question is about the PAF neuron. I saw your PAF neuron, some have 2 axons. So these are really 2 axons, or they are just because, I mean, the 2 branches, actually they are all initiated from same axon because they're close to soma. You couldn't clearly separate them. It's really rare they have 2 axons, I mean, extended from the soma directly.
I think, this is a very good question. We try to, we really try to distinguish this part. We would think there is a possibility that they originally initiated from one single axon, then they bifurcate to different parts, because the figure that we showed is really like a projection. But in a 3-dimensional view, we will be able to see that initially it's one. Yes, and then they bifurcate.
Okay, great, thanks.
Okay, there's a question from the audience in the broadcasting room. The question is, are there any examples on the interaction between other hormones and oxytocin in the neuroendocrine system?
So are there any examples, right?
Yeah,
in the
interactions between other hormones and oxytocin.
This is a very good question. We actually are actively pursuing this question. Like in the PVN, there are many types of neuroendocrine cells, right? CRH, oxytocin, TRH. So what we are studying is that oxytocin and the CRH neurons, they may well interact and to regulate each other. As I've shown to you that in anxiety models, they do have like opposite levels of these hormones, right? So they play, I guess, stay in tune. Hopefully we'll find out the answer soon. Thank you very much for the question.
Okay. If there's no more questions, we can move to the next speaker. Okay, the next speaker.
Thank you very much. Yeah. Thank you. So our next speaker is Qingfeng Wu from the Institute of Genetics and Developmental Biology of CAS. He will talk about the neuron fate diversification and specification.
So can you see the slide playing?
Yes, we do.
Okay. Okay, it is my great honor and my pleasure to give a talk in this specific symposium. And today I'd like to introduce a couple of studies in my lab about neuronal fate diversification and specification in the hypothalamus. Brain development is a very complex and precise process going through patterning, neurogenesis, synaptogenesis, and circuit assembly. In all animals with a brain, from insects to human beings, the complexity and function of the brain relies on neurogenesis, the process of neuronal generation from neuroprogenitors at embryonic stages. Neuroprogenitors in cerebral cortex initially undergo symmetric division to expand the stem cell pool and to switch into asymmetric division for generating deep layer and then up layer neurons, which is followed by the production of glial cells. The functioning of the cerebral cortex also depends on the generation of neuronal diversity, covering their spatial position, morphology, connectivity, physiological properties, and molecular features. Neuroprogenitors in cerebral cortex initially undergo symmetric division to expand the stem cell pool and to switch into asymmetric division for generating deep layer and then upper layer neurons, which is followed by the production of glial cells. The functioning of the cerebral cortex also depends on the generation of neuronal diversity, covering their spatial position, morphology, connectivity, physiological properties, and molecular features. And the neuroprogenitors appears to adopt multiple strategies rather than a single uniform strategies to generate neuronal diversity. In cerebral cortex, neuroprogenitors employed predetermined model for generating neuronal diversity. Currently, there are two prevailing hypotheses for this model. The first hypothesis suggests that the neuroprogenitor cells are homogeneous and multipotent, which sequentially generate deep layer and then up layer neurons. In either early or late embryonic stage, the fate of neuroprogenitors has already been predetermined, but their potency becomes progressively restricted. The second hypothesis suggests that neuroprogenitors are heterogeneous, and there are multiple subpopulations of neuroprogenitors which can give rise to either deep layer or up layer neurons. Other than the predetermined model, stochastic model has recently been proposed to underline the retinal development. In stochastic model, retinal progenitors stochastically differentiate into one of the 7 major cell types in retina, as revealed by live imaging in zebrafish. Stochastic cell fate decisions also occurs in adult stem cell niches, such as intestinal grapes. And throughout the brain, hypothalamus represents one of the most complex brain regions. Thus, it is fundamentally interesting to understand how hypothalamic progenitors used, just what strategies used by hypothalamic progenitors to generate an extreme neuronal diversity. And here I have to highlight that neuronal diversity in the hypothalamus is extremely complex, which contain a large number of peptidergic neurons. In the hypothalamus, there are at least 12 nuclei, each of which has different physiological functions. They regulate feeding, drinking, sleep, temperature, endocrine, autonomic system, circadian rhythm, and so on. We all know that there are many functionally important peptidergic neurons in the hypothalamus, including the oxytocin neurons introduced by Dr. Gao. However, how many neuronal subtypes residing in the hypothalamus is still unclear. The emergence of the single-cell RNA-seq technology has led to an expansive explosion of our understanding of neuronal subtypes in the hypothalamus. And recently, we combined the single-cell analysis and population lineage tracing to reveal the cascade diversified model, which underlines the generation of neuronal diversity in the hypothalamus. In more detail, radial glia cells or neuroprogenitor cells, intermediate progenitor cells, and nascent neurons along the lineage hierarchy contribute to the generation of neuronal diversity in the hypothalamus in a stepwise manner. And it is worthwhile to mention that radial glia cells in the hypothalamus can produce both ASCO1 positive and Neuroctu positive intermediate progenitors. More interestingly, ASCO1 positive intermediate progenitors can produce both glutamatergic neurons and GABAergic neurons. And this is very, very different from the cortical intermediate progenitor cells. Thus, we add a new model in this strategy list for generating neuronal diversity. And during hypothalamus development, we speculate that there might exist multiple sublineages which collectively drive the generation of hypothalamic neurons and their lineage progression. But how to determine the fate of a specific subtype of neurons remains unclear, and that is very fascinating for us. Thus, we come up with the question that what is the cellular and molecular logic operating within the lineage to specify a subtype of neurons? Despite we have already know that neuroprogenitors and neuronal precursors along the cell lineage hierarchy adopt a cascade diversified model to generate neuronal diversity. And Dr. Harkani's lab recently published a Nature paper and report a list of potential fating factors, that is translucent factors, which are very important, might be very important for the hypothalamus development. In combination with our own studies, we found that TBX3 is very specifically expressed in a subset of the neuroprogenitors and in a few subtypes of the hypothalamic neurons residing in arcuate nucleus. more importantly, genetic mutations in TBX3 can cause a disease named as ulnar memory syndrome. Thus, we first constructed TBX3 cria mouse line to label such a specific subset of the neuroprogenitors in the hypothalamus. After crossing the driver line with a TD tomato report line, we can induce the fluorescent labeling and validated the mouse line by co-staining TD tomato with TBX3 antibodies. Thus, we show that TBX3 is very specifically expressed in the acroterminal domain, the most ventral part of the hypothalamus. At 5 days after induction, we can see a large number of the progeny cells in the hypothalamus. At 30 days post-induction, we can see a clear expansion of TBX3-derived progeny cells. In striking contrast, the rats can label most, if not all, hypothalamic progenitors, which can just label the whole hypothalamus. If we label the TBX3 progenitors at later time points, like the E11 or E13, as expected, the population size of TBX3-derived progeny cells reduced, and their spatial distribution was more restricted within arcuate nucleus, the feeding center within the brain. And next, we sorted the TBX3-derived progeny cells and performed the single-cell RNA-seq. And from this lineage-dependent single-cell data set, we could identify 9 neuronal clusters, and 6 of which are peptidergic neurons. We validate that by a single molecular in situ hybridization. Within the arcuate nucleus, we found that KNDY neurons, AGRP neurons, and PONC neurons persisted to express TBX3, but GHRH neurons did not express that anymore. And to investigate the function of the TBX3 in the neuronal specification of hypothalamic neurons, we generated TBX3 conditioned knockout mice with NKX2.1 which can achieve a complete genetic deletion of TBX3 in the hypothalamus. And most of these animals died at birth, and some of them can survive until P30, but they all display a significantly reduced body size. We then microdissected the hypothalamus from these knockout mice for single-cell RNA-seq using 10x platform. And unfortunately, we only collected a relatively limited number of the neurons from this data set. Nevertheless, pairwise comparison of the single-cell data set between wild type and knockout mice still revealed at least 6 neuronal subtypes were lost in the knockout mice. And these clusters include AGRP neurons, PONC neurons, KNDY, FOXP1, and HDC neurons. To validate this data, we further performed the single nucleus RNA-seq using BMB platform. And based on this data set, we have more than 50,000 neurons and for single cell analysis. And we can still identify at least 12 neuronal clusters that were lost in the knockout mice. And in this plot, red label clusters are lost in knockout mice, and the black indicated clusters are potentially gained in the knockout mice. And the signature gene expression analysis of these clusters show that KNDY, AGRP, and PONC neurons in arcuate nucleus, as well as quite a few subtypes of neurons in mammillary body were lost in knockout mice, but GHRH neurons are still maintained. Very importantly, we aligned the single-cell lineage-dependent single-cell data set and that from the knockout mice, and found that quite few neuronal subtypes arising from TBX3 lineage were completely lost, including PONC and AGRP neurons, suggesting the importance of the TBX3 in the fate specification of arcuate neurons. But very, very interestingly, we found that the GHRH neurons arising from TBX3 lineage are retained, but HDC neurons not arising from TBX3 lineage are depleted. This allows us to propose that TBX3 loss can perturb neuronal fate in both lineage-dependent and lineage-independent mechanisms. For the lineage-independent mechanisms, we provide at least a couple of proof of concept evidences. For example, the GHH neurons arising from the TBX3 lineage are retained, suggesting the intralineage retention. And in contrast, HTC neurons not arising from the TBX3 progenitors are completely abolished, suggesting an interlineage interaction. And so this is the very novel cellular logic underlying the lineage progression of the hypothalamic neurons. We further perform pairwise comparison of the lost neuronal subtypes between our single cell and single nucleus on a sick data set, and found that KNDY neurons represent one of the most robust cell type that were lost in the knockout mice. And recently, Dr. Hakeni proposed that it is very important that in engineering of genetic models specific to neuronal subtypes is required to causally link neuronal identity to endocrine output, and this will be very important for the future studies. Thus, we determined to link KNDY neuronal identity and its fate determination with endocrine output. KNDY neurons is a key node to control the timing of puberty onset in the brain by projecting their terminals to GNRH neurons expressing gonadotropin releasing hormones into the pituitary. And what controls puberty onset is one of the 125 unresolved important questions by Science magazines. And the previous human genetic studies revealed that TBX3 mutations can cause UMS, ulnar memory syndrome. And most UMS patients display delayed puberty onset. However, the pathological mechanisms underlying the delayed puberty onset in UMS patients is still unclear. Here, we would like to link the Y neuronal fate specification with UMS. Single molecular in situ hybridization using this technology, we found that the KNDY neurons indicated by TAC2 and KIS1 neuropeptide are completely lost in the NKX2.1 mediated knockouts. However, their downstream targets, GNRH neurons, are not changed. And this model shows the loss of the KNDY neurons in the knockout mice and the maintenance of the GNRH neurons. We further used the TAC2 cree to achieve the cell type specific genetic deletion of the TBX3 in KNDY neurons. While a complete loss of the TBX3 in hypothalamus causes the death of the animals before P60, I think most of them died at juvenile stage, but all animals with TAC2 dependent deletion of TBX3 survive into adulthood for our analysis. In these animals, TKO animals, we found that the number of TAC2 expressing and KIS1 expressing neurons were reduced to 25% of the control, but not completely lost. At a later time point, we found the same phenotype. To distinguish whether TBX3 ablation causes cell loss or only peptidergic identity loss, we integrated lineage tracing with genetic deletion to permanently label KNDY deficient neurons with GFP. And using these PKOA animals, we found that GFP positive maintained cells are not changed at P0, suggesting that TBX3 deletion could not change the cell neuronal survival at embryonic stage. However, when we have looked at the P30 TKO mice, we found that the GFP positive maintained neurons were significantly reduced, suggesting suggesting the cell death at postnatal stage. Furthermore, we found that among these GFP positive KNDY neurons, just quite few, about half of them lost their TAC2 peptidergic identity. Thus, we think that TBX3 is required for the fate maintenance KNDY neurons. Loss of TBX3 just impairs their identity maintenance, including the cell loss and identity loss. And this is our linear modeling. With this modeling, we showed that the knockout of the TBX3 in the whole hypothalamus causes the complete loss of KNDY neurons, but in TKO mice, half of them undergo cell death a quarter of them lost their peptidergic identity, and only a quarter of them are retained in the hypothalamus. Taken together, we combined lineage tracing, single-cell analysis, and genetic models to indicate the cellular logic of neuronal lineage progression in the hypothalamus. And furthermore, we extend the cell fate to explore the physiological functions. In this TKO mice, we found that the puberty onset of the male and female mice are significantly delayed in these animals. And the female animals displayed an impaired estrous cycle. And also, all of these knockout female animals are infertile. They cannot get pregnant and give birth to any babies. At the molecular level, we found that the TBX3 staining shows the puncta structure in vitro and in vivo. This is the culture cells, and this is the neurons or progenitors on the brain sections. We further purified the TBX3 proteins and found that just TBX3 can undergo the phase separation. And this FRAP experiment shows the fluidic properties of the TBX3 proteins. Further purification of 4 human TBX3 mutants identified in UMS patients shows that all of these mutants could not undergo phase separation, in contrast to the control TBX3. And all of these TBX3 mutants also displayed impaired transcriptional activation of TEC3 and KIS1 neuropeptides. So this is the summary addressing the function of the TBX3 at molecular level, cellular level, and animal level. Okay, yeah. So the last slide is about is the acknowledgement. We thank many talented students in our lab, and we also thank the collaborators from Tsinghua University and Fudan University. So I would like to take any questions.
Thank you very much, Qinfeng, for this beautiful talk. So any questions from our audience? I guess I would like to take advantage of this opportunity to ask you a question here. So the TBX3 specifies lots of neuropeptide neurons, and particularly the Kingly neuron. I was wondering, for these mice, do they have this obesity phenotype? You know, you showed the puberty onset delay. What about the obesity? How does it connect it to the obesity phenotype?
Oh, obesity phenotype. Okay.
Yeah.
So I think just these animals also, I mean, just so if you use the POM3 and AGRP3 to delete the TBX3, so you can identify the obesity phenotype in POM3 mediated deletion of the TBX3, but not AGRP mediated deletion. But in our animals, we only use the TEC2 and KIS1 to delete the TBX3 from the KNDY neurons. But I think there's another Nature Metabolism paper reporting that the loss of the TBX3 in POM neurons causes obesity.
Okay, okay, very interesting.
Qinfeng, I have a question about when you said TBX3, when you remove TBX3 in this conditional local mice, and I couldn't see the KNDY neuron, but I saw most of the result from this adult mice. Did you see I mean, if you go to the very, very earlier stage, can you also see some neuron, this KNDY neuron? That's the P0, P10.
Okay. So we did not perform the single cell analysis at embryonic stage. So I think just the PBX3 can persistently expressed in a subset of the neuroprogenitors and the postmitotic neurons. I believe that in the immature nascent neurons within the hypothalamus, TBX3 are still expressed in the arcuate nucleus neurons.
Thanks.
Okay.
So regarding the specification model, you have different models. Do you think that this model particularly apply to hypothalamus, or it actually applies to the whole brain region?
Okay. Yeah, I think just in the cerebral cortex, the neurogenesis process is relatively simple, but in the some other, like in many nuclear brain structures, including the thalamus, hypothalamus, midbrain, and brainstem, I think this model can be applied to all of those brain regions. So the process of the neurogenesis in those nuclear brain structures seems to be much more complicated than that in the cerebral cortex. So I think just I expected this model can be applied to the other brain regions.
I have another question. In the cerebral cortex, people always focus on, anyways, so there are different neuropeptide plus neurotransmitter neurotype, right?
Okay.
How does this combined specification determined?
Okay, I see. Yeah. So I think just after we just identify the cascade diversifying model just in the hypothalamus, which underline the generation of the neurons in the hypothalamus, we are very interested in the fate specification or the specification of both neurotransmitters and neuropeptides. And I think just currently, based on our studies in the lab, we found that the specification of the neurotransmitters is very, very difficult to be manipulated and changed. But the expression of the neuropeptides is relatively easy. It's easier to be changed by genetic manipulation. And we also think that the expression of neuropeptides can change change the fate of the many post-mitotic neurons, including their survival and the axonal projections or connectomic properties of those neurons. So yeah, I think just that's our just very preliminary speculation.
Okay, so we have a couple of questions from the audience. So the first one is that are candy neuron TBX knockout males fertile?
No, just the female animals are not fertile.
Okay. Are there any other syndromes related to the depletion of or mutation of TBX3? Would it also affect the development of other brain tissues?
Oh no, I think the TBX3 is very, very specifically expressed in the arcuate nucleus of the hypothalamus, but not in other brain regions. So that's, yeah, I think just one of the reviewers asked that question about the specificity of the knockout mice.
What would it happen when TBX3 is overexpressed?
Okay. Yeah, I think just we tried to do that experiment. Okay, so we micro dissected the neurons from the arcuate nucleus. Overexpression of the overexpression of the TBX3 can increase the many neuropeptide expression, including TAC2, KIS1, HRP, and PONC. But when we just micro dissected the neurons from the cortex and other neurons in the hypothalamus, it seems that overexpression of TBX3 could not transdifferentiate the neurons into the specific subtypes of the peptidergic neurons. And currently, we are trying to just identify a few epigenetic barrier for that transdifferentiation.
Okay.
So I think the single TBX3 factors could not transdifferentiate the cortical.
We can't hear you actually. You are muted.
Okay.
So anyway, due to the time limit, so we if the audience have any other question, you can contact Qingfeng later. So thank you very much for this wonderful talk, Qingfeng. So our next speaker is also from CAS, is Dr. Shenjing Xu. He's from Center of Excellence in Brain Science and Intelligence Technology. He's going to talk about the interrogation of neuronal ensemble in the paraventricular hypothalamus. Let's welcome. Shenjing. I think you are muted.
Okay.
Okay,
just
yeah,
wait
good.
a minute. Yeah, thank you. Where's my PowerPoint? Could you see my screen?
Not yet. Okay, yes, we can.
Okay, great. Please wait a minute because I have a audio stimulation, so I need to share my sound for you guys. Okay. Okay, good afternoon, everyone. First, I'd like to thank the organizers for inviting me to this wonderful symposium meeting. Today, I'm going to talk about the development of camera imaging and its application in molecular and functional interrogation of the neuronal ensemble in the preventricular hypothalamus, PVH for short. The PVH is especially important for many evolutionary conserved survival behaviors, including feeding, drinking, fear, sex, aggression, even more. These broad functions create a challenge in studying how the PVH neuronal ensemble encodes these different survival behaviors. The PVH is also famous for having diverse molecularly defined cell types based on their gene expression profiles. The PVH is also sometimes described as a behavior state switchboard. Each cell type selectively influences one distinctive behavior state as a label line like this. This is certainly oversimplified, but most experiments in the hypothalamic region implicitly assume this type of label line schematic when they talk about hunger neurons, thirst neurons, stress neurons, and so on. From this switchboard hypothesis, we can infer the activity map of PVH neurons in different behavioral states, so like this. One cell type responds and only responds in one specific behavioral state. However, because of the technical obstacles in simultaneously monitoring the activity dynamics of multiple cell types across many behavioral states, we could neither validate this switchboard hypothesis nor explore other possibilities. So we stepped back and considered the three possible models describing the general role of molecularly defined cell types in behavioral state coding. The simplistic one is the label line coding, where neurons of a cell type selectively encodes one behavior state. The most complicated one is the full ensemble coding, where the molecular information is relatively useless for behavior state coding. Neuronal ensemble encodes behavior states irrespective of cell types. Neurons of a cell type may have unrelated activity patterns. And the intermediate one is the grouped ensemble coding, where neurons of a cell type have coordinated tuning responses. Behavior states are encoded by specific combinations of these cell types. This had been extremely difficult to distinguish these three models because actually they describe the relationship of molecular identity, neuronal dynamics, and neural coding of behaviors. This is a long-standing question in neuroscience. To address this question, it requires simultaneously monitoring neuronal dynamics of multiple cell types across many behavioral states in the same animal. When we started this project in 2015, there was not a suitable approach meeting this requirement. So to overcome these technical obstacles, we developed a camera imaging platform, where camera stands for calcium and RNA multiplex activity. Our goal was to link the molecular identity and the neuronal dynamics as parallel as possible. For this, we put single-cell RNA-seq, deep brain imaging, and multiplex fish together and made them work seamlessly. First, we used single-cell RNA-seq to unbiasedly classify the PVH neurons into cell types and identify their marker genes. Then we express GCAMP in the PVH neurons without regard of two cell types, and monitored their activity using 2-photon imaging through a green lens while the animal was undergoing many behavioral tasks. After all of these functional imaging experiments, we removed the brain and sectioned it. Then we found the same neurons imaging in the brain slices and in the in vivo imaging volume. Once we had this correspondence, we performed the multiplex fish with these marker genes to build the detailed molecular identities of individual neurons. Finally, we will assign the molecular identity to the neurons imaging in the in vivo imaging volume. In the first step, using single-cell RNA-seq, we classified the PVH neurons into 12 cell types and identified 11 marker genes. These marker genes comprise many canonical PVH neuropeptides like the oxytocin, where Professor Gao introduced, and the prodynorphin, vasopressin, SST, TRH, CRH, and PEG. In the meantime, to understand the activity dynamics of PVH neurons, I performed a set of deep brain calcium imaging across many behavioral states. Here is an example of volumetric imaging during a field retrieval experiment. It includes 8 imaging planes. During the experiment, in the rest state, the animal is on a treadmill. Then a leak spot will be extended. The animal will start to eat immediately, and then a field tone will be presented. The tone was conditioned with foot shock on the day before this experiment. So the animal will freeze and stop leaking. Then the tone is off, but the leak spot is still there to evaluate the fear of the animal. Finally, the leak spot will be retracted. So let's see the PVH neuronal activity dynamics in these different behavioral states. Now, the animal is on a treadmill in the rest state, and the baseline activity is low. Then spotting immediately leak, strong activity, and turn on, even stronger activity, stop leaking, turn off, no leaking. Activity gradually returns to the baseline level. Additional functional imaging experiments include eating during hunger, drinking during thirst, ghrelin-induced hunger, and leptin-induced energy surfeit. After all of these functional imaging experiments, I carefully pulled out the green lens and removed the brain and sectioned the brain, and found the same GCAMP expressing neurons in the confocal microscope. Then I found some distinctive neurons with distinctive shapes that could be confidently recognized in the ex vivo images. For instance, in this in vivo imaging, these neurons look quite different. And this one is like a side view of a foot. And this imaging is an overlay of Z-projected confocal stacks from 3 consecutive brain slices. Comparing these 2 images, we could confidently call this one, and this one, and this, and this, and these 2, and these 2. Using these distinctive neurons as starting points, we could gradually match their surrounding neurons like this. And these are the neurons in the bottom slice. Once we have these correspondences, we performed multiplex fish in the brain slices. From the multiplex fish result, we found this foot-like neuron highly expressed CRH and co-expressed MPY1R. Using this corresponding ROIs, we could extract the activity dynamics of individual neurons and their corresponding gene expression profiles. After this hard technical development, we were rewarded with this unique common imaging dataset. It contains activity dynamics of 319 neurons across 11 behavioral states, along with their gene expression profiles. Usually this dataset, if you use traditional glial method, it will take tens of people to do tons of work. So our common imaging method really speed up to link the activity dynamics and the molecular identities. Using these gene expression profiles, we classify the PVH neurons into 11 cell types. As you can see, there are a group of neurons showed consistent inhibited responses after ghrelin injection and a consistent activated response during fear. In our common imaging dataset, we can track their molecular identity. We found these neurons highly expressed CRH, MPY1R, and prodynorphin. This common imaging dataset provides tremendous information about neuronal activity dynamics, molecular identity, and behaviors. We spent about 2 years analyzing this dataset and discovered a bunch of interesting biological findings. Because of the time limit, I will just go through some of our main findings in the next couple of minutes to show the major conceptual advances. First, we examined whether the PVH neuronal ensemble showed evidence for label line coding of behavior state. In principle, label line neurons are a group of neurons defined by their functional responses. They specifically respond in one behavior state, but not in other behavior states. So we ignored the gene expression profile and exhaustively searched for the label line neurons encoding different behavior states. We brute force searched all possible activated or inhibited label line neurons. When the number of encoded behavior states was small, selective tuned neurons were evident. For example, when the number is 2 or 3, there are tens of selective tuned neurons. However, beyond 6 behavior states, there were no selective tuned neurons for each behavior anymore. This result suggested that the PVH neuronal ensemble could not encode the behaviors of these behavior states using label line coding. We also calculated the activity map of all PVH cell types in different behavior states. This activity map is apparently different from the activity map of the label line coding as I described before. It also indicates the PVH cell types encode different behavior states using combinatorial coding. So far, we have excluded the label line coding model. Common imaging allows simultaneously monitors activity dynamics of multiple cell types in the same animal. It allows us to discriminate these two combinatorial coding models. To compare these two models, we developed a machine learning algorithm to decode the behavior state using the PVH neuronal activity dynamics while keeping the cell type information or shuffling the cell type information to generate dummy cell types. If the decoding performance with cell types is greater than the decoding performance with dummy cell type, it supports grouped ensemble coding. Otherwise, it supports full ensemble coding. Now, here are the decoding results. Cell types provided a significantly higher decoding performance than dummy cell types. So these results suggest that the PVH cell types encode behavioral states using grouped ensemble coding. It also validated the use of molecular information to reduce the dimensionality of PVH neuronal ensemble for behavior state coding. In principle, grouped ensemble coding provides higher scalability and flexibility than label line coding, and higher robustness than full ensemble coding. In addition, using feature selection analysis, another machine learning method, I think Longwen also introduced in his talk, we identified the MPY1R was the most predictive gene for functional responses across all behavior states. We also found MPY1R was expressed in multiple PVH cell types. It is well known that MPY transmission is through volume diffusion. Put them together, it suggests that the MPY modulates and coordinates multiple PVH cell types, like the conductor of the PVH neuronal ensemble. In summary, we found PVH cell types encode behavior states using modulated grouped ensemble coding, but not the popular view of the label line coding. Our results also validated molecularly defined neurons are important information processing units in the PVH. It also provided an unprecedented view of the complex relationship of molecular identity, neuronal dynamics, and the neural coding of survival behaviors in the PVH neuronal ensemble. As for the coding strategy in other brain regions, our technique and the analysis pipeline provided the systematic solution for it. In the PVH project, we performed the multiplex fish in 14 micron brain slices. The section step destroyed the spatial information of neurons imaging in vivo. It dramatically increased the difficulties of common imaging at the sample preparation and in vivo, ex vivo registration. To make the common imaging user-friendly, we built a multi-fish project team and developed a new multiplex fish method in thick tissue. We called easy fish. It combines expansion microscopy and HCR. Here shows the easy fish imaging volume of these 7 genes. It looks quite beautiful. Now we can perform easy fish for more than 60 genes in 300 micron thick tissue. Then we integrated the easy fish into our common imaging platform and updated the common imaging to CAMA 2.0. Because the spatial information in vivo and ex vivo imaging volume was maintained, CAMA 2.0 provides much easier sample preparation and 10 times speed improvement in vivo and ex vivo registration. Here shows the in vivo, ex vivo, and merged image stacks. We can recognize this one as this one, and this as this. When we are going through the whole image stack, we could find correspondences of most neurons in imaging in vivo, like this, and this. So the CAMA 2.0 make the common imaging much easier than before. So besides the interesting biological findings, I like to highlight the significance of common imaging platform. It merges the traditional systems neuroscience with the molecular neuroscience. It bridges the gap from gene expression to in vivo neuronal dynamics and behaviors. It also facilitates discovering new cell types in both molecularly and functionally defined manner. It does not require genome modification, so it's applicable to any organism, especially suitable for non-human primates. It will facilitate perturbations with much more precise molecular handles. It also provides a solution for rapidly accessing functional roles for the large number of new molecularly defined cell types uncovered with single-cell transcriptomics. This work was done in Genelia when I was a postdoc in Scott Stenson's lab. So I'd like to thank Scott for his strong support and inspiration for this highly risky and ambitious project, and thanks Rini for his fantastic supervision on machine learning analysis, and thank Hui for her critical contribution in developing the multiplex fish method. I thank all of my collaborators. I started my lab in Institute of Neuroscience last September, so the postdoc position is available for candidates with background of optical engineering, chemistry, bioinformatics, and neuroscience. So if you are interested in my lab, please email me. And thank you for your attention.
Thank you very much, Shenjing. This is a very famous paper now. We shared your publication quite a few times in the JC, and I discussed a lot. So thank you very much for sharing this. So any questions? Is it possible to perform karma using miniscope in freely moving mice?
Yes, it is possible that we are doing this kind of experiment, but it needs a more restriction between the epifluorescence and the 2-photon. But you still need to do the 2-photon imaging to recognize the single cell in the miniscope. Yes, it's doable.
Okay, so
and I think the other solution is that use the famous mini 2-photon microscope from Heping Chen's lab, so you can achieve the same goal.
Yeah, because I guess hypothalamus is a bit deep, so it is a little bit difficult to use as Professor Chen's scope. Yeah.
You need a higher power. Yeah, it's doable.
I have questions in the paper. You mainly, like later on, you focus on the NPY1 receptor neurons. Actually, in one of your figure, it looks like the CRH positive neurons are involved, like in all the kind of behaviors, quite a few, at least. Yeah.
Yes. So this is quite interesting that because this is a very, very long paper, so we cannot introduce most of these. But for the CRH, it's involved in most of these behavior states, and the amplitude of the CRH responses are also modulated in different behavior states. So we think the CRH is encode the salient of the stimulant, but we plan to do the specific manipulation for this type of CRH. But because the COVID-19, we delayed the experiment. But I need also mention that this CRH is not the CRH in the mid part of PVH. So because like Da Yulin's lab, they published the CRH response, it's in the mid part, but our CRH neuron is in the posterior of PVH, and this CRH co-expressed MPY1R and PED, so it's a totally different subtype of CRH. Yes.
I have a question, at least just two questions. So, so in your study, how come the NPY1R neurons in the paraventricular nucleus dominate over other PVN neurons to control so many just a different behavior?
So because the NPY1R was expressed in most of these PVH cell types, and the receptor itself, the expression level is different. So once the NPY1R released, they will have different modulation effect. So when you compare how that this gene can predict the functional responses, it turns out the NPY1R was the most predictive gene. When we started to do this this analysis, we don't know what kind of gene is most predictive. But when we got the results, we really think it is reasonable because NPY1R is a neuromodulator receptor. So when you express some receptors, then you can really have some coordinated response for this neuromodulator.
And how about the other well-characterized receptor, MC4R?
We didn't find this in our, it's not dominant in our data set. So, but I think because MC4R is also very famous, so I think we can dig deeper for this. But in our current data set, we may miss this cell type. Yeah.
Okay. And I have another very naive questions. At times, the neurons respond to a specific stimuli or behavior state do not control that behavior, right? So how do you define the behavior encoding neurons? Just does that mean only just those neurons responding to a specific behavior state?
I think this is a very good question. When we were writing this paper, we really want to use a word or term to define this state, because we really know this state, and we think it's a behavior state. But when we check all of this paper and even the textbook, the very famous textbook in Candice, Principle of Neuroscience, they mentioned the behavior state, but no definition of behavior state. So we think maybe we still need to keep the behavior state. Otherwise, we need to come a term because it's still really defined by the behavior. So once we really think this is a behavior state, then the neural activity in that period we think is related to the behavior state and encode for that. But as we know that for the homeostatic regulation, the response sometimes may be delayed. So we also do the analysis with some delayed period, and we didn't find very consistent activity. So now then we put these consistent results in the paper. But as I mentioned that there are tremendous information related to the activity and the behavior states and the molecular identities. So we're still doing the data analysis with this data set. So I think when we have more and more of this kind of data, we may think have different view of the traditional hypothalamic coding strategy. Yeah.
Okay, great, thanks.
There's another question, Shenjing, from Wu Ping. In your model of this, you propose multi neurons project to a group of cells and contribute to one type of behavior. But how are this group of neurons assembled in the brain? Are they actually physically located in the same region as a nuclei, or just the same group of cells sparsely located in the same region?
The answer is I don't know, but from what I know and from our data set, these neurons is sparsely and randomly distributed in our field of view. And from the projectome in our institute in the Institute of Neuroscience, we also performed this single neuron projectome of hypothalamus. We think the projection of these neurons actually have some module. So it really has some organization in the hypothalamus and in the paraventricular hypothalamus. So I think they have some random distribution, but it's still in the local region. But in the relatively larger PVH, they also have some organization for the global distribution.
So, you know, like there are many nuclei in the hypothalamus, while in the cerebral cortex, there are a few nuclei. Does your model also apply to the cortex?
I think so. So, but we need to do the experiment. So, I think maybe the molecular information to predict the functional response. I think the contribution may be less in the cortex because the cortex have more plasticity, and we think the hypothalamic brain regions is relatively evolutionary conserved.
So regarding these different models, what about the labeled line model that has to be like it has been accepted for quite a few years? Like, would you like what would you think that model applies to which region?
I think it should be the input of all the sensory system. So if it's going to the brain, the information already a mixture. So and because this survival behaviors of this sensory information is related to the animal survival, I think it will broadcast to the brain regions. So if I want to see some label line coding strategy, I will go to the peripheral sensory system.
Yes, I agree. So because you are working on the hypothalamus and particularly the PVN region, it is an integration center. So, so in a way, we are probably not surprised that there are actually multiple stimuli converge to this nuclei, to different populations of neurons, I guess, right?
Yes, I think this is quite an interesting question. I think that this is in the neuroscience field. Sometimes there have some conceptual theory, and like in the hypothalamic field, people always talking about the label line coding, but in the high brain function study guys, they don't believe the label line coding. So when we first presented our work in MIT, most of them don't believe there are label line in the brain. So, but you know, in the hypothalamic field, people always talking about the label line and want to study, find some label line neurons and assign their function and assign their molecular markers.
Okay, yeah. So thank you. Okay, so thank you very much, Shenjing, for this very nice talk and that's beautiful work. So the second part will be the special talk. We are going to have 10 minutes rest break, and then our special talk, the speaker will come in. So stay in tune and have a cup of drink replenish our brain and our human body. Okay.
Thank you.
Thank you.